I'd like to welcome you to Lecture 13, which will be Chapter 11 of our book on the nervous system. And in this lecture, we're going to look at just the structure and organization of the nervous system. So to start with, you can see the extent of the nervous system. Of course, this is leaving out minor nerves, but the nervous system we're going to see includes structurally the central nervous system, and then we're going to look at the peripheral nervous system. And we're going to see, very much like the skeleton, we can divide it into two components, just like the axial and the peripheral. But we're going to also see that we can divide the nervous system not only anatomically, but also functionally throughout the body. And this seems to get rather confusing, but just keep in mind that there's going to be some overlap in these different classifications of the nervous system, what we call both the anatomical structure of the nervous system and the functional structure. So we can see here that there are going to be three functions of the nervous system. We can divide the nervous system functionally into what's called sensory input, and this just means receiving signals from the environment and from the body, basically just baseline information. This is not going to include any interpretation of the information. So for example, light, dark, color, or sound are all information coming in. We're going to see that integration is more of the interpretation of the sensory input. And this means, how does your body perceive this? And this is very important because this tells us why some people feel more pain than others, and basically why certain people see things that other people don't, and how we experience the sensory information is very unique to the individual based on upbringing, environment, and again, structure of the nervous system. So the integration center is more or less, how do I interpret this information to use to carry out an action? And the action part of the nervous system we're going to call the motor output, okay? And things called effectors are going to be in charge here. The more common effectors we're going to talk about in this class are going to be muscles and glands. Sensory information later we're going to call affective information. Affective means basically incoming or emotion. So be aware of the terms affector and effector, because effector is outgoing information, affector or affective is incoming information. So when we look at this whole idea of the structure of the nervous system, we can see again there's affective or sensory input, integration, which involves interpretation of that information. This is where different parts of the brain, we'll learn later, give their input to the information that's coming in and actually interpret it. And part of that integration we're going to see in the spinal cord is also going to be involved in something called the reflex. And then there's the motor output. That means, what am I going to do with this information that's coming in the way I interpret it? So as discussed a little earlier, we can see that anatomically we can divide the nervous system into the central nervous system, which is the brain and the spinal cord, and also some regions that we're going to call the integration and command center areas, because we can actually see these anatomically as we look into the central nervous system. And then we have the peripheral nervous system, which is made up of paired, what we call spinal and cranial nerves. So these are branches that come out of that central nervous system, either through the brain case or through the spinal cord, and their job is going to be to carry messages to and from the central nervous system. So again, to show you the anatomical distinction between these two, is there's your central nervous system with your brain, which is your main integration center, and then the spinal cord, which we're going to see is going to be more of what we call a basic reflex integration center. Okay, and then there's your peripheral nerves, which come out of the base of the brain. You're going to see a little later, and out from each of the spinal regions. And we're going to see that this kind of divides the body up into sections. And it's really kind of interesting when you look at these structures here, and this is going to be very important to pay attention to. When you look at the arm is reserved to certain nerves that come out of the spine. The same is going to be true for the lower appendages. So when you think about how the nervous system branches out, think of you having your legs split out and your arms spread out like this to understand these neural pathways. So these are all what we call segmentation. The body is segmented into particular neural regions that physicians can isolate and look at various things, various conditions and diseases of the nervous system. So the peripheral nervous system has two major functions. Again, we're going to see the sensory or afferent division and the efferent or motor division. So the peripheral nervous system's main job, again, is to convey information, unlike the central nervous system, which is mostly going to be interpretation. So in the sensory component, we're going to see what we call somatic fibers. Somatic means these are impulses that come from the skin, the skeleton, the muscles, and the joints, and basically send signals to what is going on. Signals that include pain, touch, stretching, and generally body position. And then there's going to be what's called the visceral afferent region, and this contains impulses from the organs. That means your internal organs. So there's two separate signals here we'll be seeing, signals that more or less convey the environment and body position, and those signals that convey more of stuff that's involved in general metabolic homeostasis. And then when we look at the motor part, again, these are just general impulses that listen to this information and then do something about it. So let us hone in a little on the motor division of PNS, or the peripheral nervous system. So again, this is to be looking at somatic, what we sometimes call voluntary nervous system, and this is under conscious control for the most part. Now, be aware of what conscious control really means. It means that you just have an important override of this. Now, that means this gives you the ability to lift something and pay attention and say, I want to lift something up. When you're walking up and down the stairs and holding posture, there is a little unconscious control. That means involuntary control. That means the brain and spinal cord are working real hard to keep you out of the loop to think about it. Okay, so really we have command over a lot of things, but there's many other things in this somatic system that do go on, particularly associated with posture, balance, walking, and doing certain activities. And every now and then you hear about muscle learning and things like that. And this, like when it comes to dancing or playing the piano, that's not necessarily muscle learning. What that is that it's all these what we call reflexes that are part of the somatic system that's basically training muscles sometimes to get away from your brain and not think about particular activities, but put them on what we call the reflexive level. That means you can do it without thinking about it. Now you can start that motion, but a lot of reflexes do a lot of the fine coordination that you don't have to think about all the time. So we can somewhat see the somatic systems. You can see that there's nerves coming out of the cervical area. Now pay attention to this. This is something called a plexus. This is a group of nerves that do fine control, particularly over your upper appendage, over your arms. And this is very important to see how all these nerves interconnect to allow your arm to do these incredibly complex movements, okay, at the command of your brain. We can see here there's a more simpler arrangement. When we get down to the lower back and to the sacrum, we can see again a big complexity. And so these little units you see here, these big, what we call a plexus, know that term, are interconnected areas that allow you to do these incredible movements associated with balance, associated with control, and just general body movement. Because the upper appendage and lower appendage have much more complex movements than the torso and the lower back, the back. Now, the motor division of the peripheral nervous system also includes what we call autonomic or involuntary control. So what we're looking at here now is we usually delegate this to more of what we call visceral motor nerves. Okay, now when we say autonomic, it means that you can control this, but it takes incredible training to do this. Some people are a little better at it than others, but primarily these are things you don't want to think about a lot. But again, there's kind of like an overlap between these two in a way, depending, and this is to be due to your integration center, how your integration center works, because the integration center allows you to have free control over certain things, no control over others, and partial control over other things. And but when we look at the autonomic nervous system in general, mostly what you have here is the integration center is taking more control that it doesn't delegate to you. So this is a system that controls smooth muscle. That means the digestive processes, the heart, your cardiac muscle, and it stimulates the secretion of glands. And there's going to be two subdivisions we're going to see of what's called the autonomic nervous system. Now think of the autonomic nervous system as output. That means information going out to the body. That is sensing the environment, it's sensing the internal organs, and what it's doing is sending out information for how your body responds. So we're going to see that there's two branches, what's called the sympathetic and parasympathetic, and these are more kind of functional components when we look at the nervous system. So can we actually see the parasympathetic and sympathetic nervous system? Yes, we can. So here we go. This is your peripheral nervous system, and pay attention to where these come out of. This is very important. Okay, so when we look at, again, this autonomic control, these are nerves that parallel some of the other peripheral nerves. Remember that. So there's other nerves that go into the vertebrae and out that are paralleling these. This is just another layer of nerves. This is what makes neurosurgery and understanding the nervous system in general very difficult, because you have all these pathways that run into each other, overlap, and you have to learn how to distinguish these. And believe me, they're not color-coded like you see here. So when we look at what's called the parasympathetic nervous system, this is the nervous system that's associated with what we call rest and digestion. Okay, and we can see where it comes out of. The parasympathetic nervous system usually branches out of what we call the cranial nerves. That means they come right out of the skull, not the cervical vertebrae, but right from the base of the skull. And a lot of these openings you see, these foramen you see at the base of the skull, okay, are not just for blood vessels, but also for these parasympathetic nerves. And we're going to see that they're going to be involved in various processes that literally spread throughout the body. So just because a nerve comes out of the base of the skull doesn't mean it just goes to upper body parts. We're going to see it goes right down to, the lower parts of the digestive system. The other part of the parasympathetic nervous system comes out of the sacral area, your sacral vertebrae. And these are mainly going to be involved in sexual response and also urination, which is why the two of these are actually linked in a way. So what does your parasympathetic nervous system do? What are some examples? Because this will be asked on a test. Is the parasympathetic nervous system constricts your pupils. That means it makes the pupil, the opening of your eye, really tight, really closed. It stimulates saliva. Think about that. This is involved in digestion. It slows the heart rate. That's involved in rest. It constricts airways. That means it makes the airways a little less likely to take in gas exchange. It stimulates stomach activity, because this again is associated with digestion. It helps to regulate the liver, okay, in the uptake of glucose. And it helps to stimulate intestinal activity. Now, when it comes to the sacral portion, what we see here is the parasympathetic system contracts the bladder. That means this is involved in urination. And this is a problem a lot of people have. When they're not relaxed, it's very difficult to urinate, because it's difficult to contract those muscles. And even though this is an autonomic response, we can have some brain control over that response by being stressed. Okay, and this could also be true for defecation. We'll see a little later. Parasympathetic nervous system promotes erection and also sexual response in the female. And this is very important, again, when we look at the effects of stress on reproductive system, reproductive performance. So let's go on to the sympathetic nervous system, which is sometimes called the fight or flight, or in some cases, stand still part of the nervous system. So we see that what this does is dilate the pupils. Now, why during the fight or flight response do we want to dilate or open that eye up? What that does is it allows a lot more information, what we call peripheral information. It opens up the eye so they can see a lot more versus constricting the pupil. Constricting the pupil is more for close vision. This is more for distant vision. So it gives you more of a visual acuity. Now, this is sometimes when you're under stress, it's very difficult to focus on near stuff because the pupil is dilated. Now, the pupils also dilate and constrict due to light and also due to drugs and other things. So this is a very generalized comment I'm giving you here. There are many factors, both reflexive, autonomic, and also conscious control that affect pupil constriction or dilation. Now, sympathetic nervous system counteracts the parasympathetic in the case of salivation. Okay, sometimes under stress, particularly physical exercise stress, we get what's called dry mouth. That's because you're not producing saliva. This increases the heart rate. And actually, when we mean by increased, the heart naturally has a very high heart rate without control. The sympathetic nervous system more or less regulates that high amount to maximum to rather high. Parasympathetic technically really slows the heart rate down from the original, fast beat. Sympathetic pathway relaxes airways. That means it allows a better exchange of gases in and out because that helps to make breathing, much more functional, and also helps it to correlate its function with the heart rate. Okay, we're going to see this is also involved in vasculature, in blood pressure. Okay, sympathetic nervous system inhibits digestive system activity. That means in the stomach, the digestive system, and the liver. And what's neat about it too, and you don't see this with the parasympathetic nervous system, the sympathetic nervous system has control over some of the endocrine system, particularly with the production of what we call epinephrine or norepinephrine. And these two hormones, or also they're called neurotransmitters, help to stimulate the stress response, and they kind of facilitate the increase of heart rate. They also make the nervous system a little more acute and muscle action a little more sensitive. So when you are running, you have a little more sensitivity there. When we look further down now, we can see that the sympathetic nervous system relaxes the bladder, okay, and it promotes, now this is funny, it promotes ejaculation and vaginal contraction. So there's actually two separate responses that are involved in sex. You have to have a parasympathetic or resting component, and then you have to have kind of like a stress-related component. And one basically follows the other. So you have to have a little relaxation and then eventually a stress response. And the stress response comes from actually hormones released during the sexual response that promote sympathetic or kind of fight or flight activity. So now let us kind of review the division of the nervous systems. And so we see the central nervous system. Here we go with pulled apart and teased apart. And we can see coming out of there are what we call somatic sensory fibers, and this one is going to the skin. That means that signals from the skin are going in directly into the spinal column. And here we see sensory information, visceral information is going directly into the spinal column. And then out of there from the central nervous system comes motor fibers that control either glands, okay, or in this case, skeletal muscle. And then we can see also coming out of there are what we call sympathetic motor fibers and parasympathetic motor fibers that help to control particularly the heart and the urogenital system. So that's your central. Now let's look at the peripheral branch. And the peripheral is divided into sensory and motor. And again, these are just fibers that come out all along from the cervical, again, down to the lumbar, that are involved in information going into the body. So it's taking information, and guys, the information is very true as it goes in from particular body regions. It's all segmented so the brain can tell whether something is coming from a pinky or coming from your big toe or coming from, the middle finger versus another finger. Now, sometimes there gets to be a little confusion there because when body parts are close, particularly in the appendages, it's very difficult for the brain to sometimes sift out all that information. And sometimes we get this phenomenon, what's called referred pain. That means the pain feels like it's in one area or not another because the incoming signals are sometimes confused. So that's your sensory information. Then the outgoing or motor information involves what we call somatic and autonomic nervous system motor information. The somatic, again, is going to be voluntary structures like organs, okay, and muscles. And then there's the autonomic, which is going to be your visceral mostly. So we're going to be controlling internal body organs. And then that is interpreted into sympathetic or parasympathetic. That means, is that information I'm sending out going to be either due to the relaxation response or the stress response? So again, just memorize this. Understand how these work. The nervous system is very complex on how it is subdivided. And the central nervous system, peripheral nervous system, each play roles, okay, in the information that is coming in, the information that is going out, and how it's interpreted by the body. So let's look a little about the structural histology of the nervous system, because later we're going to go into how neurons work. So there's two principal cell types in the nervous system, because understand that a nerve is basically a bundle of neurons, we'll see. So when we talk about nerves, you're not talking about nerve cells. You're talking about hundreds, thousands of nerve cells working together. So what does an individual nerve cell look like? And could you tell about them? What could you tell about their job? So we see here is what's called a bipolar neuron or nerve cell. And a bipolar nerve cell, we're going to see, is very involved in integration. And you can tell by this, what they have is incoming information and outgoing information. You're going to learn that this part here is going to be either putting out information or putting in information. So this is taking information from a lot of sources and putting out information to a lot of receiving areas. And these are usually sometimes called interneurons. These are sometimes involved in conveying information and also interpreting and controlling information. They're very much involved in reflexes. That means how you blink your eyes when someone puts a fist towards your face, or when you fall, how you put your arms out without thinking about it, and also how your eye sees motion is all due to these bipolar or what are called interneurons. And then there's what's called unipolar. And don't try to make sense of that, but bipolar basically means there's two poles coming out, two ends to it. This has one end right here that comes out of that cell, unipolar. These are usually sensory. And again, look at the information going in, the information going out. Sensory neurons sometimes take in the same type of information from different areas and then distributes it out. These are very rapid neurons that their job is to mainly get that information in and where it needs to go. And then is what's called multipolar. Now notice the poles. There, 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 and there. A pole just means one particular end, little notch that you can see on there. And from that notch comes something. That's a pole. That's how you remember this. So with the multipolar now, you're going to learn that they take information in, and this is taking information in from different directions. That means that this booger right here is probably listening to other neurons and sometimes overlapping neurons. So he's taking information again, from different areas, usually in what we call a neural pathway, and then it's putting it out into a general area. These we're going to see are usually going to be called motor neurons. Most of them are involved in taking information, particularly sensory information in, or other motor information in, and putting it out for one specific function. And this is the problem sometimes when we're frightened, is sometimes you get a lot of this information in, and the information is competing, and this poor part doesn't know what to do, and you seize up. So these are involved a lot in muscle movement, again, gland secretion, taking a lot of information in, put it into one major area. And then there's what are called pyramidal cells, because they're shaped, the body's shaped like a little pyramid, and sometimes the whole cell is shaped like a pyramid. So with pyramidal cells, these are usually found deep in the brain, and they're going to be major integration neurons, very much like the bipolars, but much more complex. And this is where we find a lot of complex reflexes in the brain and also memories taking place. Okay, the other cells that make up the nervous system, besides the neurons, are what are called neuroglia, or sometimes glial cells. The names vary depending on where you learned them. So we're going to look at cells called astrocytes, because they look like little stars, microglia, which means small cells, ependymal, we'll see what they are, oligodendrites, and that means few processes or arms, satellite cells based on their location, and what are called Schwann cells, actually named after, I don't know if he was German or Austrian, but named after a researcher whose name would be pronounced Schwann. Astrocytes, and we'll see a picture of them in the next slide, okay, are the most abundant of the glial cells. And we're going to see that they work together with other glial cells, and particularly with neurons. Think of glial cells as assistants to the neurons. So what astrocytes do is they're very abundant. They cling literally tightly to the neurons, particularly around, we're going to see the ends of the neurons, and they're also going to be associated with capillaries that feed the neurons. We're going to see that they support and brace the neurons, and they determine how much food and waste come into and out of the neurons. They're associated with what we call capillary permeability. Don't worry so much about that. But these are the caretakers of the neurons. They help to feed them. They help to remove waste. We're going to see that they also guide them through the body. Astrocytes are very important in nervous system healing. That means if you pinch a nerve or damage a nerve, the neuron, if it's still alive, can regrow as long as the astrocytes are in place. So you don't want to lose these glial cells, even though they are capable of replicating. In some cases, they have to be there initially to help repair damage. And again, they're involved in growth of the body, we're going to see, and the placement of how the nervous system forms in the embryo. Like I mentioned earlier, they control the chemical environment for a neuron. That means they control not only nutrients, but also potassium, sodium, calcium, very much like we learned with muscle tissue. And we now know that they participate in information processing. We now know that they could actually help to regulate memory and our control of memory and placement of memory using various signals, including ATP, the energy. So now we can see what the astrocytes really look like and what they do. So that here's the body of the astrocyte, and this is kind of what gives it the star-like appearance. Don't worry so much about that. I mean, microscopists can see this, maybe we can't. But look what that cell does. There's your neuron. So they basically wrap around parts of the neuron. Okay, they totally enclose blood vessels. So when you look at this, this is what's amazing about the central nervous system, is that they are a barrier between the blood and that neuron. The neuron is not in the central nervous system, is not in direct contact with the blood. Okay, so look at this. This is amazing what we see here. That means that nutrients that come from the blood have to pass through and into the astrocyte before it gets into a neuron. And this is a way of kind of filtering the blood and protecting the neurons from any bad things, not all bad things, but many bad things. And this forms a system, what we call the blood-brain barrier. Blood-brain barrier, because it's a barrier between the blood and the brain. So certain drugs will not pass into the neurons. And this is very difficult, particularly when you start looking at, how do I deliver, certain medicines to the neurons. Some of these medicines are blocked, but usually it's a way of protecting many toxins, not all toxins, from getting to the neurons. What else we see here is that materials from inside the neuron wastes pass into here and then into the blood. So these astrocytes also act sort of like a way of helping to remove waste and facilitate the removal of waste from the neuron. So this is incredible system we find in the central nervous system that heavily protects it, but also can work against it sometimes. And the decay of the astrocytes can leave us open to a lot of infection and a lot of toxins that can get through the body, through the blood. So now we're going to look at the microglia, and we're going to see that they're almost related, at least functionally, not so much structurally and developmentally, to white blood cells in a way. They're very small, obviously. We're going to see that they're kind of more ovoid, that means more round, not as angular as regular glial cells. And we're going to see that they migrate towards injured neurons. They're going to be a very important part of that healing process. And again, you could fix a damaged neuron. You cannot replace a dead neuron. Neurons cannot replicate. I mean, there are rare instances where this might be able to occur, okay, but this doesn't occur usually naturally in humans. And in many animals, like birds, they can replace neurons, particularly neurons in our brain. We can't. What we try to do is repair as much damage as possible and rely on kind of having other neurons pick up for the job of a lost neuron. So they're going to be responsible for helping to repair neurons, but they cannot replace a neuron. And we're going to see, just like white blood cells, they phagocytize or eat microorganisms and neuronal debris. That means they clean up the waste. And this is very important in your nervous system, because as you get older, it's evident that microglia might not be able to do their job as well, them and the astrocytes. And this causes an accumulation of waste products in the brain cells, usually fatty type compounds and residual protein type compounds that can cause dementia and eventually death or loss of function of the neurons. What do microglia look like? Okay, here they are. Look, there's your ovoid body, and they have all these appendages that kind of touch the neuron, and they act again as a protector of the neurons, and they also go around ingesting material. So they help to remove waste from the neurons. They work together with the astrocytes, but also remove debris and potentially harmful chemicals that are in the solutions. Because remember, this is all filled with interstitial fluids. Now, the ependymal cells are slightly different than what we've seen before. These are not so much associated with neurons in the central nervous system as the previous cells we've seen, but they mostly line parts of the central nervous system, particularly the brain and much of the spinal cord. Their job is to basically remove fluids from the blood and put them into their own fluid-filled cavity that cushions and acts as a nutrient source and a source of sending signals, chemicals, and collecting waste from the central nervous system. So this is more found in a sac, and we'll see this as we look more into the brain, into what we call the ventricles, and a small opening we see passing through the spinal cord. So these ependymal cells produce a cushioning protective fluid that's also a nurturing fluid. Anything that gets into this fluid can be hazardous for the body, because this is separate from the rest of the body, and it's very difficult to fight infections inside this fluid-filled cavity. And sometimes you'll hear in children that are born with too much or too little cerebral spinal fluid, because this affects the cushioning of the brain, and also it can also inflame the brain or shrink the brain, and cause some problems. Then is the oligodendrites. And these are the equivalent of astrocytes, except they're found in the central nervous system. So these are sort of like the astrocytes of the central nervous system. And they form a type of material called myelin in the brain. Okay, now, what do they look like? This is it. They are just sheaths of cells. So what this cell does is it wraps around, usually we're going to call the axon of the nerve fiber. So they wrap around neurons, individual neurons. And we're going to see that they act like an insulator, particularly to expedite a nervous signal. So now when we look at the satellite cells and Schwann cells, as we pronounce them, satellite cells are sort of like the astrocytes are to the central nervous system, but these behave that way for the peripheral nervous system. So they're the little guys that clean up. They help to form kind of like sometimes a barrier between the blood and these cells. And then the Schwann cells we're going to see are very much like oligodendrites. And don't worry about neurolemmocytes. This is whoa. It just means that when you look at a peripheral nerve cell, you don't see the nerve cell for the most part. What you see are the Schwann cells wrapped around it. So these surround what we call nerve fibers, neuron fibers, and form these sheets that helped to make for faster neural communication. Sometimes as we get older and in certain diseases, these Schwann cells die down, they lose their function, and the nervous system becomes very delayed and slow, which can be a problem. This is what they look like, and I want you to notice something here. Look at the gaps now. These gaps are going to be called neurons of Ranvier or nodes of Ranvier. We'll cover a little later. Okay, and we're going to see that this helps to cause what's called a saltatory nerve response. We're going to see that when a nerve sends a signal across this thing, the nerve signals kind of jump along the whole length of this thing. The Schwann cells we're going to see speed up nerve signals. And not all neurons are covered with these. Many of them are, but some are not, because you don't necessarily need Schwann cells. But there's your satellite cells, there's your Schwann cells. So now let us look at more detail about the neurons, because this is where we're going to spend most of our time in this course. Is neurons, understand, do not replicate. They do not undergo mitosis. They literally are long-lived. They are there with you for life. And believe me, they do wear out. There's a good point to not replicating in that you don't lose their function in the process of them replicating, and also they don't shorten their telomeres. That means they don't show age-related damage that's associated with mitosis. But the problem is they do show age-related damage because they can't sometimes repair themselves. They can't repair their DNA easily, and they cannot repair a lot of biochemical damage that can be repaired during the mitosis process. So they do age, just like skeletal muscle. They do age, and in adult humans, there are a few, if any, stem cells that can produce neurons. And again, many birds, many types of animals, okay, like us, can replace neurons, but we can't. So again, they're amitotic with few exceptions. Neurons tend to have one of the more highest of the metabolic rates of any cell next to muscle. So neurons and skeletal muscle particularly eat a lot of your calories. They have a plasma membrane that has an electrical charge to it. We're going to see the cell promotes an electrical charge in this membrane, and we'll learn about that in the next lecture when we look at nerve cell physiology. And neurons are very much involved in what we call cell-to-cell interactions through neurotransmitters and through direct electrical signaling. So what does a neuron look like? Okay, and what are its parts? Okay, understand that neurons don't just work individually. They work in bundles. or what we call tracts, depending on what you're looking at. So in the central nervous system, bundles of neurons working together are called tracts. Okay, and bundles of neurons working together in peripheral nervous system we call nerves. So when we look at a typical neuron, whether it's central or peripheral, they all have one common feature called the nerve cell body. The nerve cell body gives life to the neuron. It controls the metabolism, provides ATP, and also it produces the neurotransmitters, the chemical signals that help one neuron communicate with another through what we call cell-to-cell communication. They have a region called dendrites. This is where information comes in. So these are sensory components, if you want to think about it that way. This is where information comes in and either goes directly to the nerve cell body or sometimes directly into this part here. Like in sensory, we see this in sensory neurons. There's your nucleus, and don't worry about the nucleolus. And then this structure here that comes out of here and runs through here, notice the Schwann cells, is called an axon. This again is actually part of the neuron. It's a long tube filled with cytoplasm and cytoskeleton. And this is where the signal that is coming in goes in one direction from here to this end here. So we're going to see that information in a neuron is always going to travel, okay, from the dendrites all the way to the end here, which we'll name in a minute. So in many neurons, we find glial cells wrapped around them. In this case, in this neuron, this is a peripheral system neuron because it has Schwann cells. These are neuroglia cells, remember, and they leave these little gaps called nodes of Ranvier. Okay, we'll look at the significance of that in a minute. And then at the end, we have what's called the axon terminus. Some people call it the bouton because it looks like a tiny little button under the microscope, and it's from the French word bouton. So this is what conveys information from one neuron to another or to a structure that this neuron is going to control. So to reiterate, the flow of information through a neuron is going to be into the dendrites, either from another neuron or from a sensory structure. And then the information goes out through what's called the axon terminal, which is a secretory region. It produces chemicals called neurotransmitters from what are called these terminal branches. And these terminal branches can go to usually one, but sometimes one or more, but very few, what we call effectors. That means this can produce a secretion, or it could go to a gland, to a muscle, or it can go to another neuron and just convey information. So understand the parts: the dendrites, the body, the axon, which starts here and runs all the way through, and then the axon terminus, which contains most of the time what we call terminal branches. Okay, and there's your Schwann cells, and neurolemma is just a fancy term for nerve cell membrane. The nerve cell membrane, the plasma membrane, is called the neurolemma in this case. And don't worry about Nissl bodies or nucleolus. I mean, these are just secondary structures. They are important, but we're not going to memorize them. So equally important to the function and structure of a neuron, again, and we mentioned these earlier, are the Schwann cells, which produce this covering. It's a fatty type covering. It's basically just cell membrane with almost no cytoplasm in it that's wrapped around the axons. And we call that a myelin sheath. And it's not found on all nerve cells. We see that some nerve cells have their own myelin and others don't, but they associate with other cells that have myelin, and they almost share the myelin in a way. And then the neurolemma, okay, is going to be the nerve cell membrane, but particularly referring to the little parts that form the gaps between the Schwann cells. And we'll come back to this when we look at nerve cell function. Okay, so when you see it, again, just to go a little more in detail about the Schwann cells, is that they wrap multiple times, okay, around an axon, okay, and these neurolemma areas are going to be what transmits what we call the action potential, particularly at the nodes of Ranvier. So the nodes of Ranvier is basically going to see electrical communication goes on that transfers information from the dendrites all the way to the terminus. So that's the importance of the nodes of Ranvier, is they help to allow that communication to be one way down a neuron. This is a close-up of the Schwann cell wrapping around an individual axon, okay, and this would occur during either healing of nerve damage or in a fetus. The Schwann cell wraps around and wraps around till it's tight and it's almost all squeezed together and becomes almost like a complete its own little uh, 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 um, nerve cell, its own little membrane, almost acting like its own little membrane around the nerve cell. Now, I discussed earlier, we have two types of neurons. We have what are called myelinated and unmyelinated. Okay, and here's your myelinated right here. Notice they have myelin, and what happens is we'll see when a nerve signal, that means information coming this way and going that way, passes through a cell like this, it basically jumps across what's called these nodes of Ranvier. And it's just like you, basically, let's say going into an airport and you see these little moving sidewalks and you get on them, these little moving sidewalks speed you along. Now, in an unmyelinated neuron, what we see here is that a potential kind of walks across this rather quick, but not as quick as this. Why do you want nerves like this? Sometimes we do have slow acting neurons, but mostly when we see unmyelinated neurons, they're sitting next to a myelinated one and sometimes borrow the nearby myelin. This is the way that the body saves on resources. We can produce less Schwann cells and get double to triple the purpose out of them. Something you're going to hear about, read about in your book is what's called axon thickness. Our body has axons of different thicknesses depending on where that neuron is and what that neuron does. And we can classify these into what we call axon types. Uh, um, we have three types of A and then a type C. And don't worry about the classification scheme of this, but we have A alpha, A beta, A gamma. Okay, and it just has to do with a way of designating the thickness, because what we've learned was that the thicker the axon, the faster a neural impulse can pass through it. Okay, we see many creatures that have very rapid nervous systems. They have very thick axons. Okay, but one way of enhancing an axon is not just thickness, but again, also putting Schwann cells around it. So what happens, we see when electricity passes through the inside of an axon, that narrow part of the axon. This is the outside of the axon, that's the axon, this is the fluids inside, is when we get an electrical stimulus is determined by the flow of materials in that axon, the flow of sodium and potassium. When there's a wider axon, you get more of a rapid flow than when there's a narrow one. So this produces an impulse that requires much more resources, but is a little quicker than that. So thickness, we're not going to worry too much about, but do know that there's several ways we can speed up or slow down a neural signal to help build and coordinate the body, is by the thickness of the axon or by the myelin sheath. Now, another thing you hear when learning about the nervous system, particularly the central nervous system, is this concept called white and gray matter. And actually, it's not white or gray. It really does look that way when you put it in formaldehyde. And then the formaldehyde interacts with fats and proteins in the tissue and makes it appear white or gray. Really, it's more of degrees of kind of pinkishness, okay, and one that looks kind of glistens one that looks kind of flat. So the white matter technically is all the myelin and all the glial cells, which are very high in fat. And the gray matter is mostly going to be neuron cell bodies and unmyelinated fibers. So this is mostly, so gray mostly refers to the communicating cells. The glial cells are mostly the white matter. And you can see here the gray right here and the white. So it's not so much gray and white as much as kind of like more of a shiny and a dull when you look at real brain tissue. And you can see here that's a ventricle of a brain. This is a transverse section of a brain, and this, you can see this is the this looks more like the middle part of brain, because you can see what's called the temporal lobes right there. Okay, so this is your ventricles. The ependymal cells will be here, and you'd have nerve cell bodies throughout here and axons running through here. Now, some other terms you're going to hear. These terms distinguish how we name groupings of cells in the central versus the peripheral nervous system. And you've heard a couple of these terms before. So when we have so what happens in the nervous system is neurons and glial cells don't act alone. They are bundled together in large communication pipelines that allow hundreds to thousands of them to work together in unison or separately within a bundle to control the responses, to control how much of a response or where that response is going to go with it to a particular effector that it's working with. So we see the term nerve again. This refers to collections of neurons outside the central nervous system, in the peripheral nervous system. Now, neurons run in tracks. That means nerve cells are all lined up together like this. That's the body of the nerve cell. That's the axon, all going in one direction of information from dendrite to terminus. These are bundled together in what's called a nerve that makes up what's called in the peripheral nervous system. Now, these nerve cell bodies all lined up together is called a ganglion, and you'll see them as knots in the nervous system. You'll see these in your specimens and in pictures. Now, when it comes to the central nervous system, and this is looking at a transverse section of the spinal cord, that a transverse section of the brain, we have what are called tracts. That means tracts are bundles of axons within the brain that go from one region to another and convey very integrated information. There's a nerve tract right there, the optic nerve, from the eye, I should say from the brain to the eye, and also from the eye to the brain. And then we see these structures called nuclei, which are bundles of nerve cell bodies. That means they are connecting information, sending out information, receiving information, and we find them in the brain and in the spinal cord. So understand, you'll hear the term nucleus. Don't confuse it with the nucleus of a cell. These are bundles, again, of nerve cell bodies. These are like mini brains. Think of a ganglion and a nucleus as a mini brain. They are interpretation coordination centers, except these are more in the peripheral nervous system. These are in the central nervous system. So you have mini brains throughout your body. Now, what is actually a nerve? Because it's very difficult to see the structure of a tract, because tracts are very complex. Sometimes they're very small and difficult to actually isolate. So a nerve, that means this is part of peripheral nervous system. A nerve, okay, is basically bundles of neurons. Remember muscle tissue. Muscle fibers are bundles and bundles and bundles. So what we see when we look at a nerve, it is going to be basically a bundle of axons with or without myelin, all bundled together like a pile of spaghetti. It looks like right there, dry spaghetti. And each axon is going to be covered by its own little connective tissue covering called an endoneurium. This is what maintains the chemical environment around an axon, just like this is true for muscle we saw. So the endoneurium is going to wrap around the Schwann cells and the neuron. And then all this bundle here is surrounded by a perineurium. That's called a fascicle, very much like in muscle. And then these fascicles are going to be glued all together and wrapped up in what's called an epineurium, which is what makes a nerve. And one nerve can go to one body organ, and then what happens is these fascicles can either go to one particular region or spread out, depending on what type of control you want. This is a cross section under the microscope of what a nerve bundle looks like. So here we go. There's your perineurium of a fascicle, and you can't see really the epineurium because it's real large, and your endoneurium is right about there. And don't worry about the longitudinal section. This gets very confusing, and I'll never show this to you under a microscope. Okay, so again, now let's focus again on the jobs of the different neurons that you saw. So we have three major types of neurons that we're going to look at in this class to understand their function and to understand what are called neural pathways. They're going to be what we call the multipolar, okay? And remember, these are going to be what we sometimes call motor neurons. What they do is they take a lot of information and funnel it into generally one area. There's the bipolar, and we're going to see that these are generally going to be taking a lot of information from one area and putting a lot of information out. These are going to be usually are going to be conveying information, distributing information, and kind of figuring out what to do with stuff. And then there's the unipolar, which are going to be usually sensory. That means they are taking information in from many areas and focusing it into one fast-acting region. So let's look at these again. Here's your multipolar. Lots of areas where the dendrites come in. That's where the multi comes from. And these can come from basically sensory structures. This information come from other neurons. What he's doing is listening to a lot of information and funneling it right in there. Okay. There's your bipolar, and these are going to be neurons that control information. It's going to direct information. So it takes a fair amount of information coming in, puts a fair amount of information coming out, but the main thing is that they're going to be integrators. That means they're going to be associated with these guys in getting information from one place to another and sometimes controlling the direction of that information and coordinating things. And here's your unipolar. Notice there's your pole, and you have what looks like a giant axon there, which in a way that is information comes in and goes directly through. The nerve cell body is not interacting with this. Okay, so this information is usually coming from basically some type of environmental signal or from an organ, and this information is then going to the central nervous system or to another integrating region that's going to take that information and somehow get it to the brain or get it to where it needs to do its job. Here again, we see Purkinje cells. Okay, these are found in the brain. Look at those dendrites. That's one bad hair day. So this takes an incredible amount of information and notice funnels it into one area. And this is sort of how these are a lot of times associated with memory. There's a pyramidal cell, again, found in the brain. There are extreme types of multipolars, okay, and there's different types of bipolars, and there's a unique type of a unipolar, okay? So don't worry about the weird examples, but do know that there are subcategories of multi, bipolar, unipolar, which sometimes these are their own unique cell in themselves. Okay, so again, the functional classification of neurons. To review, sensory or afferent, motor or efferent. That means these are taking information, and that information better come in sometimes from one source, from a lot of sources, but it has to transport quickly through the body. The motor, what this is this information either comes out quickly or slowly to affect something, to make a gland secrete, to make a muscle move. The speed all determines on where they're placed in the body and what exactly they're associated with. And then the interneurons, they do what they call shuttle signals. That means they direct them, control them, and sometimes literally act as a little mini brain in themselves. So let us look at some locations of the different neurons. We're going to see there's a motor neuron that is peripheral, but it does branch out of the spinal cord. And this motor neuron gets information from either from other regions of the spinal cord or from the brain, and it sends a signal to a muscle to make a muscle contract, or it can make a gland secrete, depending if it is somatic or visceral in its job. The eye has many bipolar neurons, which these particularly help in you identifying large objects and turning them into one, and particularly in seeing movement, interpreting movement. And then we have sensory or unipolar neurons that take information from receptors and put that information into the central nervous system. So next we're going to look at more about how nerves communicate through each other in what we call neurophysiology.